welcome to everybody, and thanks for joining us again for another Evenings at Whitney. It's great to see such a large turnout, both on Zoom and here in person. So tonight, it's my pleasure to be introducing you to Dr. Robert Lamb. And Robbie is a research professor here at the University of Florida. He's actually based in UF's Nature Coast Biological Station, which essentially, if you don't know, it is Whitney Lab's sister station over on the Gulf Coast. As well as that, he's in the UF School of Forest, Fisheries and Geomatic Sciences. He originally obtained his bachelor's degree in Oregon State University. After that, he traveled to Ecuador to obtain his master's. He then returned to the US to study for his Ph.D. at Brown University. And then he moved on to do a postdoctoral position at Woods Hole Oceanographic Institute. So he's pretty well traveled. And he studies a wide range of topics. Essentially what they all have in common is fish, as you'll hear later. And this includes everything from fish ecology from a community perspective, with specializations in distributions and biogeography, food webs, population dynamics, changes in response to climate, and behavior as well. So he's a pretty well-rounded researcher. And he's spent most of the past 10 years working in far-flung places such as the Galapagos Islands of Ecuador, the Gulf of Maine, and also throughout the Caribbean as well, and has done a number of studies throughout Latin America generally. So he's pretty well versed with tropical and subtropical fish species as well as those further north. And impressively, his work has been used in fisheries management, and it's also been crucial to the setting up of some marine protected areas and designing them and making sure that they're being put in effective places. And it's been featured in prestigious media outlets such as the National Geographic and the New York Times as well. So I'm delighted that he's willing to join us here tonight. And he's going to give us a talk entitled Fish Ecology in a Changing World, which I'm really looking forward to seeing. So welcome, Robbie, and thanks for joining us.
Hello. Hi. Hi, everyone. Welcome. And thanks, Dave, for that wonderful introduction. Thanks to both Dave and Mark Martindale, the director of the Whitney Lab, for the invitation to give this talk today. And thanks to all of you for coming out and joining us this evening. It's a real pleasure to tell you about the research that I've been doing. As Dave mentioned, most of this work is going to be talking about my experiences in the Galapagos Islands and coastal Ecuador. I am a researcher here at University of Florida working in Sierra Key, and we've got some projects that we're getting off the ground right now that kind of tie into some of these themes. But this is an opportunity for you guys to see kind of how I think about fish ecology and how it's really an issue of importance in our current changing world. So why study fish communities? Well, fish communities actually are a perfect model system for studying ecological change. They are the most diverse vertebrate group on Earth, with tens of thousands of species described. They provide food and livelihoods for billions of people worldwide, mostly around coastal areas. And when we look at species, when we compare across species and across scenarios, we can actually get quite a bit of insight into why some species have mechanisms of resilience and others decline for the same type of climate perturbation. So some of the questions that I'll be addressing in this talk today: how do marine protected areas affect fish communities? We know that marine protected areas, otherwise known as marine reserves, are an important tool for fisheries managers. How do they work? What are the effects of extreme environmental conditions on reef fishes? So this ties into ocean warming, but also changes in wave stress and wave induced water flows. And finally, I'm going to talk about the research that I did as a postdoc scholar at Woods Hole, which is looking at how food web structure varies with upwelling strength, which is the delivery of nutrients to the surface ocean, and temperature, because we're looking at how temperature and with a view forward to climate change might affect fish communities. So to look at this first one, how do marine protected areas affect fish communities? The main issue with marine protected areas is that you basically remove the effect of fishing. And this was the first undergraduate research project that I did as a student at Oregon State University. You can see me there bushy-eyed and young student working in the Bahamas. What I was doing in the Bahamas was looking at when this Exuma Keys Land and Sea Park was put into place, we wanted to understand not only how does this affect the species that are targeted by fisheries, such as large sharks, but how does that effect propagate throughout the entire food web or throughout the entire fish community. And what we found was that if we have the presence of a marine protected area, the Exuma Keys Land and Sea Park, what we're looking at on the left side here is what is the effect, the reserve effect on different fish based on the size of the fish in question. So each point here is a species of fish from the Bahamas, and if it's positive, that means that the reserve had a positive effect on that species, and if it's negative, it had a negative effect on that species. And what we see is that the largest fishes, such as sharks, large jacks, large groupers, are positively affected by the marine reserve, but that there's actually an inverse effect on intermediate-sized fishes. So these are like gag grouper or coney. These intermediate predators are actually declining in the presence of large predators. And towards the tail end of this, we see that the smallest fish kind of have a neutral effect. But we think of this as kind of a size-specific effect of the marine protected area, that the largest species that are directly affected and targeted by fisheries can benefit from the fishery removal, and that has cascading effects throughout the ecosystem based on the size of the species in question. So not only do marine protected areas have impacts on the fish targeted species, but those effects propagate down through the food web. In New England, where I've been working since 2014, we're trying to get protection for this place that you see towards the top there, Cassius Ledge. In fact, we presented two different locations, Cassius Ledge and the one down here, which is Coral Canyon, New England Coral Canyons and Seamounts, to the Obama administration in 2016 as potential protected areas under the Marine Monuments Act. Now, the type of information that we were collecting to establish this marine protected area was we were doing censuses of fish species. So this is cod on the left, pollock in the middle, and cunner on the right. And these are color-coded by Cassius Ledge. That's that isolated seamount. It's about 80 miles off the shore of Portsmouth, New Hampshire. And then the purple ones are coastal areas that are comparative sites along the coast of the Gulf of Maine and northern Massachusetts. So what we saw was that there was up to 600 times higher biomass of these fish species at Cassius Ledge relative to fished areas along the coastal Gulf of Maine. And in fact, what we think could be happening is that, especially for cod, which is a heavily overfished species in the Gulf of Maine, this could act as a source population that could seed populations elsewhere in the Gulf of Maine, and that it really requires protection in order to facilitate that replenishment. We just returned this last summer, and we're going to be there again this summer because, as I mentioned, there were two locations that we originally proposed, Cassius Ledge and the Coral Canyons, New England Coral Canyons and Seamounts. The New England Coral Canyons and Seamounts are about 200 miles offshore, and as you can see, they're very deep. They're off of Georges Bank, so they're about 1,000 meters deeper or even deeper, whereas Cassius Ledge comes up to about 40 feet from the surface, so it's a submerged seamount. Now, fishermen continue to like to use Cassius Ledge. They utilize it for the lobster fishery, whereas the Coral Canyons and Seamounts really are not being accessed for any fishery purpose whatsoever. If you present two different locations, one of which has a significant negative pushback by a significant proportion of the community and another one doesn't, what's going to happen? They're going to give you the one that doesn't have any pushback. So the New England Coral Canyons and Seamounts is now a Marine National Monument thanks to this work. We're still working on Cassius Ledge, and in particular what we're trying to do is work with the local fishing community there to establish what are the requirements that we would need to be able to get community support for this marine protected area. Like I said, we're continuing to do censuses out there. So we actually now have data since 1987, 2014, which was when this study was taking place, and we just returned in 2023. So we're seeing two things. One is that there's a consistent increase in cod and these other fish species at Cassius Ledge relative to coastal sites, and also that those trends are changing over time. We're seeing an increase in pollock abundance over time and an increase in cunner as cod has decreased since the 1980s. What we think in particular in the case of cunner, that middle species, is that it's actually replacing cod ecologically as cod is fished out. because Cunner has a pretty similar ecological niche. They feed on a lot of the same benthic invertebrates, whereas pollock is an open water schooling species. We think that the increase in abundance of pollock is actually more tied to ocean warming in the Gulf of Maine. The Gulf of Maine is one of the fastest warming bodies of water in the world. So again, I think that doing this like species comparison, comparing across species and looking at how different species respond to the same pressures, whether it's climate, fisheries, or what have you, is a really powerful approach. So another MPA where I've been working, and this will be kind of dominating the rest of my talk, is the Galapagos Islands. Now the Galapagos Islands are located about 800 miles off the coast of Ecuador. They're right on the equator, so they have the maximum amount of tropical insolation and energy from the sun year-round. Now, most places that are on the tropical ocean do not have a lot of nutrients, because what happens when you have a lot of sunlight and warm water on the surface of the ocean, it forms this lens, this warm water lens, that depresses the thermocline, which is the transition point between warm surface water and cold deep water, to below about 60 meters. That means that the nutrients and oxygen that are in deep water don't get up to the surface very readily. So that's why in tropical oceans, coral reefs are very important for primary production, for photosynthesis, because corals have photosynthesizing algae in their tissues that get nutrients from the corals, and the algae provide carbon to the coral. It's a mutualism. Now in the Galapagos Islands, where we also have tropical sunlight, there is a cold water current that comes from the west and hits the west side of the archipelago called the equatorial undercurrent. This current comes up to the surface from about 300 meters depth and brings with it cold, nutrient-rich water to the surface. You combine cold, nutrient-rich water with tropical sunlight, and what you get is one of the most productive marine ecosystems in the entire world. And obviously, the Galapagos has long been known for its hallmark as one of the places where Darwin learned about evolution and developed the theory of evolution. There's a lot of endemic animals that only live in the Galapagos, so from a conservation perspective, it's a very important place. It's also interesting ecologically because the Galapagos formed from a hotspot similar to the Hawaiian Islands, which means that there's this point of magma coming up out of the Earth's crust, and the plate moves over it, and so new islands emerge on the west side of the archipelago while older islands are carried towards the continent of South America. And those islands formed in the middle of the ocean very far from any sources of colonists. So new species arrived to the Galapagos Islands from 3 different regions. We have species coming from the coast of Chile and Peru, and that ecosystem, which is dominated by the cold Humboldt Current, which comes up from Antarctica, is very cold, very productive, and brings a lot of nutrients up to the surface. In contrast, species that come from the Indo-Pacific, from the west side of the Pacific, are coming from an ecosystem that's very low in nutrients, very warm in temperature, and so these are tropical species versus subtropical or temperate species that came from the coast of Peru and Chile. And we also have species that come from Panama and Mexico, which is a different biogeographic region. So all these species are now coexisting in the Galapagos and are experiencing the exact same temperature and food regimes. That gives us the context to then compare how do these species respond to the same type of perturbations such as climate change or fishing. So with that context, I've been working in these 12 sites since 2014, and every 6 months I go to each of these locations. I do censuses of the reef fish community, and we also take oceanographic measurements to determine how are ocean conditions such as temperature changing and make correlations with those changes in populations. So we do this by doing underwater censuses and 50 by 5 meter visual transects. That means I swim that 50 meters and I count visually every single fish on 2.5 meters on either side. I identify each fish to species, and I even estimate the size of each fish so that we can get an estimate of biomass. So it's a lot of counting. Thank you. The Galapagos has about 450 species of fish, and about 12% of those are endemic. That means that they are only found in the Galapagos Islands. These are 2 undergraduate students that worked with me when I was at Brown University, Calvin and Maya. They are now both in Ph.D. programs at UC Santa Cruz. No relation, they just both ended up there. But what they're doing here is a CTD cast. So with CTD, that's conductivity, temperature, and depth. And what we're looking at is how much the water column changes as you go down in depth. Like I mentioned, the thermocline. At what depth does the thermocline exist? and also to what degree is water being mixed by waves and currents. And we also put out these loggers that are taking measurements of temperature every 10 minutes throughout the year at 6 and 15 meters depth. So we have really good measurements of what the water conditions are like throughout this period. This is another undergraduate student that worked with me in the Galapagos, Marissa Agrawal, and this is a study that she published, or that she has in press right now as part of her undergraduate thesis. And I mentioned, looking at the thermocline, this is what these thermoclines look like. As you go down in depth, you change in your temperature or dissolve oxygen. And what she was looking at is this habitat-forming ecosystem engineer, which is a black coral. These are not corals that photosynthesize, but rather are capturing food, planktonic food, using filter feeding apparatus. And they're really important because they're one of the only large upright forming corals in the Galapagos that provide habitat, three-dimensional structure for other things to live in. And what she was looking at was, to what degree does differences in upwelling strength predict the depth at which black corals exist? And what she found was that you get an increase in the probability of occupancy, or the probability of occurrence of a black coral, as you go down in depth. So these are deep water affiliated species, and that different sites varied in the depth at which you could get black corals up to the surface. So we think that this is an indicator of to what degree you get warm water being delivered up to the surface, because these species are very sensitive to that temperature condition. So that's just one of the studies that we've been working on. When I first started in the Galapagos in 2013, I didn't know this, but we were actually on the verge of a very strong El Nino event. These El Nino events are marked by an increase in temperature and a decrease in the amount of primary production in the surface waters of the Galapagos. So this is comparing 2013 to 2015 in the same time of year. 2013 was a La Nina period, which is when you get a strong upwelling of deep nutrient-rich water to the surface. You can see that the difference from average temperature, the temperature anomaly, was very cold around the central or the tropical Eastern Pacific, and you can see that point where the Galapagos is off the coast of Ecuador, very cold water. In contrast, during these El Nino events, you get about 5 degrees centigrade positive sea surface temperature anomaly. That's very, very warm for the ocean in Galapagos. And that means that, again, you get a lens of warm water on the surface that decreases primary production and increases the metabolic rates of all these species because they're living in an aqueous environment. So that leads me to this next question, which is, what are the effects of these extreme environmental conditions on reef fishes? And I'll talk to you about these El Nino events and also about wave energy. So one way to measure the strength of an El Nino is the Oceanic Nino Index, or ONI, and it's basically similar to that anomaly. So a +2 is about 2 degrees centigrade above normal for that time of year, and a -2 would be about 2 degrees centigrade below long-term average for that time of year. And you can see kind of the history of these El Nino and La Nina events over time. The 82-83 El Nino event was a before-after kind of moment in the Galapagos. There were several species of coral, of algae, and one species of fish that were endemic to Galapagos that have not been seen since that 82-83 event and are believed to be extinct. 97-98 was another strong El Nino event. I remember growing up in Oregon that the 97 El Nino was a very strong one. The Willamette River actually overflowed its banks. So this is a phenomena that has impacts all around the Pacific. And then the latest one was actually we're in the middle of an El Nino event right now, but before that was 2015-16, which is the event that I'll be talking about. So what does an El Nino mean in the Galapagos? Well, oceanographically, there's two things. The first is a drop in primary production, which is the amount of new biomass being generated by plants in the ecosystem. So we measure that through satellite imagery, and we have a proxy, which is chlorophyll, the pigment that plants use to photosynthesize. And you can see that throughout the archipelago, there was a negative chlorophyll anomaly during this period. And the other thing is an increase in sea surface temperature. So if you're on the west side of the archipelago, which is where that cold water current hits directly, that's where the highest temperature anomaly was, positive temperature anomaly existed, about 5 degrees centigrade. And throughout the archipelago, we had these positive temperature anomalies. So remember, we have got a depression in the amount of food at the same time as an increase in the metabolic rates of species that are ectothermic, that are responding to the temperature around them. One of the first things that popped up in the 2016 El Nino event was an ulcerative skin disease. And you can see that how this manifests is it first starts off as like these white sort of patches on the skin. and then progresses to scale loss and the exposure of bloody subcutaneous tissue. It's really ugly. And when we first encountered this in 2016, we were doing our normal censuses, and we came up out of the water with everybody's eyes just huge, like, oh my God, did you see that? Those fish had some crazy ulcers. So we quickly worked with the Galapagos National Park to try to figure out what's going on here. We collected a bunch of fish. We did tests for histopathology. We did genetic analyses. And we know that this disease is now recurring in the current 2023 event. So these two images on the right are from this year. And I actually have an NSF board right now with colleagues from UF Fisheries, Don Berenger, Roy Yanong, and Kuti Subramaniam, and we're working on this disease again because we don't quite have it pinned down to what the causal pathogen or the environmental conditions that are required to trigger the outbreak. We know that it has to do with El Nino, and again, the combination of high temperature and low food availability. But what exactly causes that disease outbreak, we're still trying to figure out. So hopefully we'll have some information on that in the coming year. And like I said, we're doing this again. We were just in the Galapagos last month. There's Roy working on a diseased fish. We're also starting to expand into ectoparasites. So during these warm water events, there's an increase in the load of ectoparasites as well. Can't really see it that well, but this is a gill parasite that we pulled from the gills of one of the king angelfish that we caught. Sometimes they'll actually be like packed in there, and it's hard to believe that these fish can even really survive with so many parasites. And like I said, we know that this is related to water temperature. So warming oceans have both parasite load effects and disease outbreak effects on these fish. I'm really dedicated to incorporating undergraduate students into the research that I do. These are two undergrads that I have right now at UF. That's Camila Koros-Balev on the left and Melanie Gomez on the right. Camila's been doing a study that looks at the effects of the pandemic-induced reduction in dive tourism in the Galapagos on fish behavior. So because of the pandemic, people weren't allowed to go to the Galapagos for about a year, and it went from about 20,000 visitors every month to 0 for about 10 months. And so we were curious, we went back to places that have a lot of dive tourists normally, and we did a behavioral study on cleaning interactions. So hopefully Camille will be publishing her research in the next year or two, but she's a really stellar undergrad student. And Melanie, what she's going to be working on, and she's doing right here with another undergrad, is stable isotope analyses. So I'll be talking about that in a little bit, but basically what that allows us to do is trace food webs and how food webs are interconnected. And so Melanie's going to be looking at the effect of El Nino events on these food webs. A previous undergrad student that I had at Brown did another study looking at the effects of these El Nino events on benthic communities. And in particular, she was looking at these cyanobacterial mats, which you can see up on the top. And during these warm water periods, cyanobacteria can spread over the reef really quickly. In fact, we saw up to 30% cover of the cyanobacteria. That's a lot. That's 30% of the entire benthos covered by these cyanobacterial mats. And they kind of smother corals and other things, so it's really kind of a noxious thing that nothing really likes to eat. And she published this study in Marine Ecology Progress Series. We couldn't keep her in science. She's a stellar athlete, so she's actually a triathlete for US triathletes, but we don't judge her for that. Another undergrad student, Sofia Castello-Tiquel, recently published her undergrad thesis with me. She was looking at sea star distributions and feedings in the Galapagos Islands. So what we did here was we looked at the 6 major species of sea stars. How are they distributed throughout the archipelago, and what do they feed on? And so what we're looking on the left here is a multi-dimensional plot that kind of compresses all of the different sources of food that each species consumes. And then it's color-coded based on each of these species of sea star and what they eat. And you can see there's quite a bit of diet overlap, but there are different breadths, like niche breadths. So Mithrodia, the purple one, has the broadest niche breadth. It consumes the most different types of food, as compared to Pentocerastor, that's the blue one that's got the most reduced diet. And so we can kind of get at how much different species have similar ecologies and make predictions about how they might respond to a perturbation like climate change. And we also looked at how they were distributed throughout the archipelago. What we found was that sites that are terraced, so these are sites that are in black, have the highest abundance of sea stars because they have mixed habitats. They're basically like stair steps. And so there's vertical walls where they can hang out, but there's also sediment that accumulates on the benthos in the flat areas, and they feed on that sediment. So yeah, I'm really proud of these undergrad students, and it's something that I hope to continue doing here at University of Florida. But returning to this ENSO El Nino story, so again, this is the Oceanic Nino Index. We're looking at kind of a reduced temporal spread here, but we can clearly see that 2015-16 El Nino right in the middle. But it wasn't just an El Nino event that we covered. We actually had a pre-El Nino, then an El Nino, and then 2 La Nina periods. So again, La Nina is when you get a resumption of trade winds, a resumption of upwelling, a big pulse of food into the ecosystem. And so we wanted to see, well, not only what is the effect of the El Nino on fish populations, but the convex, or both sides of this, what is the El Nino and the La Nina effect, and how does that vary among species? If we compare all the different species in the Galapagos, what traits tell us how a particular species will respond to this environmental variation, and is that generally applicable? So I told you a little bit about those different sources of biogeographic origin of the species that coexist in the Galapagos. I really thought that that was going to be the whole story. I was like, the Peruvian species, they're pre-adapted for the cold, they'll do really well during La Nina and poorly during El Nino, and those Indo-Pacific species, which evolved in a high temperature, low productivity environment, would probably do even better during an El Nino. It turned out that had nothing to do with it. And in fact, it all has to do with what you eat. So in terms of your diet, you can make really clear predictions about population dynamics in terms of how susceptible you are to change during an El Nino or La Nina based on what you eat. So what it comes down to, what I've plotted here is the abundance in the top and the biomass in the bottom of all species grouped by trophic category. So what do they eat? And you can see 2 pretty clear trends. One is that there was a decline across groups in El Nino of 2015-16, and an increase of most groups in the La Nina that followed. But this is very clearly constrained by what you eat. And we can see the biggest oscillations are for the planktivore species. So they appear to be the most closely tied to oceanographic variation. And so this suggests that it's not a limitation that's directly tied to physiology, because we know a lot about how different species have different thermal tolerance windows. Some species are adapted to warm conditions or adapted to cold conditions, but none of these species appear to be close to their limit. In fact, what appears to be the case is they are all dependent on a particular food resource, and the degree to which that food resource responds to this oceanographic variation predicts the population trajectories of the fish that rely on that resource. And so we know that plankton has these boom-bust cycles that tie to oceanographic variation with El Nino, La Nina, and planktivores follow those same boom-bust cycles on about a 6 month to 1 year lag. And you can see that there's different trends with abundance and biomass. We get these spikes in abundance about every year, but they're kind of at the same season each year. And so those are recruitment pulses. Those are new small individuals recruiting to the reef, and then they kind of get eaten, and so biomass goes down a bit, and then there's another pulse of recruitment, and it goes back up again, and then they get eaten, but some of them grow. And so we're kind of like coming back to this like stable equilibrium that it was before that El Nino in 2016. So yeah, again, I really think that this comparative context is a really powerful way to look at this. And again, what we're kind of coming away with from this is that we have El Nino associated decline that's magnified in obligate planktivore species, followed by La Nina-driven recovery via larval recruitment, and then growth of those larvae. So I said that I was going to talk about 2 different sources of environmental variation. One was temperature, another is wave exposure. So waves in the Galapagos Islands primarily come from the south because of storms in the Southern Ocean during the austral winter. And waves are an incredibly powerful and important hydrodynamic force affecting all organisms in the ocean. And part of my thesis was looking at, if you look at these islands, this is Camaño on the right and Las Palmas, which is just a little peninsula on the left, you can have an exposed side facing the south with prominent swell, and a sheltered side of that same location that are completely different ecosystems, separated by less than 50 meters. And so it appears to be completely driven by wave exposure. So we really wanted to understand this, this pattern, and just so you can see kind of what it looks like, locations that are sheltered from waves have really, really high densities of these pencil urchins, and the sea floor you can see is basically a cement of crustose coralline algae. It's this grazer-resistant algae. There's almost no upright or filamentous algae whatsoever. And then wave-exposed locations have, you can see there's this like green fuzz everywhere. That's filamentous and foliose algae that's growing up. And you can even see that around the edges of the cracks and crevices, there's again this crustose coralline algal pavement because urchins are living in those cracks. They kind of wedge themselves up into the cracks using their spines. And then in lulls between waves or between periods of high waves, they can kind of come out of the crack, feed on that fringe, and then go back down. So we know that we've got these urchin barrens in low flow environments and crevices in high flow environments with really, really high fish and algal biomass. At Las Palmas, which was one of the two sites, this is a reef map where we actually stitched together a whole bunch of images to make like a large image. And you can see each of these little black dots are urchins. So that's upwards of 60 urchins per meter squared. It's a really, really high density. So obviously these urchins are foraging down all of the available algae in this ecosystem, and that prevents fish from being able to live there because there's literally no food for them. So we wanted to understand this a little bit better, and we put out exclusion cages where we had a fish access. They could come in from the top, but urchins were excluded by the wall. We had an urchin access that had sideless cages where the urchins could come in from underneath, but the fish were excluded by wall and roof. We had a treatment that was exposed to all of the herbivores, so there was no caging whatsoever. And then we had this full herbivore exclusion cage. And we compared these in high exposed and protected areas and in 2 different locations. And what I did was I put out these, what I call ulva sandwiches. Ulva is a type of algae that's very palatable. Everything eats it. It's like lettuce, sea lettuce. And I take 2 pieces of aqua mesh, which is used to make like lobster pots, sandwich them together with the ulva sticking out in the middle, and that kind of keeps it pressed together, but the fish can access it and pull pieces out. And you can pre-weigh that, deploy it in your experiment, and then weigh it again, and you have like the biomass consumed in the intermediary. So we put that out in each of these different exclusion cages in high and low flow environments. Here you can see me kind of installing them, and there's this whole like group of urchins just waiting for me to leave so that they can go ahead and eat it. So it was a pretty cool, experimental manipulation. And what we found was that so these are the 2 locations, Camaño and Las Palmas, and then we have exposed and sheltered habitats. And these are the 4 different exclusion treatments. So the minus all, that's the full cage, neither fish nor urchins have access. Plus all, they both have access. Plus fish, only the fish can get there. Plus urchins, only the urchins can get there. So when we compare the effects of urchins to the full exclusion, what we see is that in sheltered habitats, urchins have a significant effect on biomass. Like they reduce the available biomass almost all the way down to 0 relative to those exclusion controls. So in sheltered habitats, urchins are the dominant grazer. However, in these exposed habitats, there's no significant difference from those full exclusions. So urchins have a negligible effect as herbivores in high flow environments. In contrast, fish kind of showed an opposite pattern. In the exposed habitats, they had a significant effect on algal biomass, and in the sheltered, out of one of the 2 places, they had a significant effect, and the other one they didn't. We're not really sure why they weren't feeding in that location. I think it had to do with high water turbidity, and they kind of, if they can't see very well, they feel like they might get eaten by a predator. But in the exposed locations, we had significant effects across the board. So this suggests that fish are utilizing the ecological resource of algae that's available because urchins are excluded via waves. So the exclusion of one group, of one guild of consumers by a physical force, wave pressures, allows another group to feed and carry out its ecological function. And I think sort of kind of conceptually, this is how I look at it. If we look at mean flow speeds, so we actually measured in situ those flow speeds, and the proportion of algal biomass removed, and we look at all of our experimental manipulations, these lines are just sort of like general trend lines, and the dots are the actual data. We see that urchins dominate the herbivory guild at low flow speeds, less than 28 centimeters per second, and that fish dominate at high flow speeds above that. But even at a certain point, fish are also excluded. So there's a certain point of flow rate and you guys have probably felt it if you're out swimming in high wave environments, like there's a point where you can't not swim against that speed. And the same is true for fish, even though they're better swimmers than us. So at a certain extreme point, you lose herbivory by fishes as well. And we actually dug a little bit deeper into this. With the urchins, there's really just the one species, but there's many different species of fish that we observe feeding on the benthos. And so we were curious, to what degree do different fish species vary in their ability to forage in these high flow rates? So what I did was I put a current meter, which measures water flow speeds in 3 dimensions once every second. And I put video cameras out to film foraging on the benthos. So for every single bite, I have a simultaneous flow speed at which that fish took that bite. And we can plot out all the bites in a sort of a histogram of flow speed. And we partition those out by the different species, and what we can see is that there's a real clustering of some species can feed at high flow and other species can't. So parrotfish up at the top, Scarus, and a couple of the damselfish species, so Stegastes bibi and Stegastes archifrons, are kind of these low flow feeders. They wait for lulls between waves and then they can go down and take a bite, but then the next wave comes and they're kind of blown away. Some other species like the lowest one, that's Prionurus laticlavius, that's a surgeonfish. If you think about a surgeonfish, it's kind of shaped like a CD, like a disc. And so if it's facing into flow, it's very hydrodynamic. And if it turns down to take a bite on the benthos, it's also still pretty hydrodynamic. Compared to a parrotfish, which looks like a football, if it's looking into flow, it's very hydrodynamic. But the second it turns, it's exposing a lot of surface area for drag, for it to be blown off the rock. And it does, you need some amount of time to look at the rock and decide, where am I going to take a bite? And if you take a bite of something good, you might want to take another bite. So there are some species differences in their ability to forage at high flow rates. We modeled that, so we actually sort of resampled randomly 1, 2, 3, 4, or 5 species from this observational data set and plotted how much herbivory occurs if you change the number of species in the community. And what we're seeing here in these different colors is if you had only 1 species, 2, 3, 4, or 5 of herbivorous fishes, how much herbivory would you expect to see across this flow rate regime? And what we see is that the difference between the lines is maximized at intermediate flow speeds. So what this tells us is that if you're in the lowest flow condition, 1 species does the work of 5. And if you're at the highest flow condition, no species can forage. So the effect of diversity or of richness is negligible at very low and very high environmental stress intervals. But if you look at that intermediate area where there's moderate flow speed, that's where the role of diversity is at its maximum. So it's this intermediate diversity effect. Or diversity has its maximum effect at intermediate environmental stress. And I think that could be a generalizable claim. While we were doing this work, we were putting out these exclusion panels or exclusion experiments and stuff. And what we noticed was if you look kind of from a bird's eye view in these sheltered locations where the urchins are grazing heavily, you see little patches of green. And what those patches of green are territories of territorial damselfish. These are fish that actually actively garden the algae in their territory. And they do this for 2 reasons. One, for food, they eat the algae. And secondly, as a form of reproduction, it's basically, if I can keep this patch of algae green and lush, despite all the herbivores that are out there trying to feed on it, I can probably also protect your eggs. So why don't you go ahead and put them right there? So it's both food and reproduction. So another undergrad student, Maya Zeff, who's now at UC Santa Cruz, wanted to ask, we have several species of damselfish in the Galapagos. To what degree are they partitioning the available habitat and food in that environment? Because being sister species from the same genus, they probably have similar ecological requirements. And there's theory within ecology that 2 species cannot completely overlap in all of their requirements because one will outcompete the other. So what we did was look at these 3 species and compare their depth distribution and also what are they actually gardening within their territory. So we found that these different species exhibit pretty clear spatial partitioning with depth. So on the left, the giant damselfish that has the most shallow restricted distribution, really you don't see them much below about 5 meters. The yellowtail damselfish gets a little bit deeper, but it's still pretty restricted to those shallows. And then the ringtail damselfish we found all the way down to 20 meters. So this suggests that there are some limiting conditions that do not allow the giant or the yellowtail damselfish to persist at depth, that the ringtail damselfish doesn't have that same constraint. When we looked at their territories, we found that, again, this is a multi-dimensional ordination plot where we're trying to compress all of the variation into 2 dimensions, 2 axes to visualize it. And basically what this means is that 2 points that are farther apart from each other on the plot are more different than points that are closer together. And what we've done here is we've plotted the community composition of the territories from the 3 different damselfish species and controls. This is just without a damselfish present. We find that the ringtail damselfish has the most diverse or variable diets. So it's got the biggest polygon of sort of dietary space, and that it overlaps somewhat with the ringtail, with the yellowtail and the giant, but there is actually quite a bit of separation. So this is what we would call niche differentiation. They're eating different things, and they're actually fostering different types of algae in their territories. So this is kind of an example of propagated diversity. So there's a diverse group of damselfish, and by virtue of there being multiple species of damselfish, they support a diversity of algae. So there's more algae present, more diversity of algal species present than if there was only one species of damselfish cultivating just one type of algae. So this has led to a whole new line of research that we're kind of delving into now. These are fish that we actually collected across a depth gradient. So this is the same species collected at the same site from 30 meters, 20 meters, and 10 meters depth. So you look at that and you look at their distribution. So there's more shallow. This is the one on the right. There's more shallow, fewer deep. They're bigger shallow, less large deep. So again, this is multiple lines of evidence suggesting that there's something at depth that's missing for this species. And I think it's probably food. This is what we call a facultative herbivore. It eats algae, but it doesn't have to eat algae. It can kind of eat other things like plankton. But algae becomes scarce as you go deeper and deeper because light gets filtered out by the water, and so there can't be much photosynthesis. So we think that they can exist at depth, but they don't do as well. And that undergrad student that I showed you, Melanie, is actually going to be looking at their inner ear bones, their otoliths, which form rings just like trees do, and we can age these fish. I think it would be interesting to see, is that smaller fish younger, or is it the same age but it only grows to a smaller size? And so this is work that I'm developing with collaborators from Chile, La Universidad Católica de Chile, Alejandro and Vladimir, his PhD student. So these guys, you can tell, they're not doing typical scuba diving. These are rebreather systems. So this is what's called closed circuit rebreathers. When you exhale, that air gets funneled back into your system. The carbon dioxide that you exhaled gets filtered out. They have a small bottle of pure oxygen that injects oxygen back into that mix, and you breathe it back again. So this has 2 advantages. One is that you don't let out bubbles, so you don't change the behavior of the fish that you're looking at. And secondly, you can stay deeper for longer. So these guys are going to be doing this kind of work with me, getting down to 45, maybe even 60 meters depth. And what we're trying to address is this deep refuge hypothesis. This is a hypothesis that if the surface ocean warms up, as it is continually doing, and it does during these El Nino events, and species in the surface ocean are outside of their thermal optimum, could they migrate to deeper waters where it's colder? And this is a big open question right now in marine ecology because it could be sort of an escape from climate change. But there's a lot of things that change with depth: light, pressure, food availability, habitat availability. So it's a pretty big open question that we're just starting to scratch the surface of in the Galapagos. So the last angle of questions that I'd like to talk to you about is, how does food web structure vary with upwelling strength and temperature? And this comes from an observation at the global scale, which is that as you go from the equator, which is at 0 degrees latitude, towards the poles, temperature decreases, right? It's warmer in the equator, colder in the poles. And you actually get a decrease in the abundance and diversity of herbivorous fishes. So from like quite a bit to 0. Like there's literally not a single herbivorous fish in Antarctica, nor in the Arctic. So that's led to the hypothesis that there's actually a thermal constraint on herbivory among ectothermic fish that have the same temperature as their environment, possibly because it's just metabolically limiting. You cannot get enough energy from a low protein source of food at cold temperatures. So in the Galapagos Islands, we have a really strong gradient in temperature that we could actually test this hypothesis with. Because some of the complaints or complaints you could call it about this study was that, well, there's a lot of things that change when you go from the poles to the equator, right? Differences in biogeography, differences in currents, differences in the evolutionary origins of species. So there could be a lot of other things that explain this trend. But in the Galapagos, we have a very condensed window of temperature variation across which we can test some of those predictions. So we wanted to ask, do fish diets vary predictably with temperature? We would expect an increase in the relative reliance on algae with temperature if that hypothesis is true, and a decrease in the relative reliance on plankton as temperature goes up. And then we also want to look at, does fish community composition vary predictably with temperature in a similar manner that it does at latitudinal scales? We would expect an increase in the abundance and richness of herbivorous fishes with temperature, even within that reduced microcosm. So for this work, I collaborated with Simon Thorold, who's at Woods Hole Oceanographic Institution, and we use this compound-specific stable isotope approach where we basically separate out individual amino acids using a gas chromatograph, which separates out molecules based on their weight. And then we look at, for each amino acid, how much of the carbon in that amino acid is called heavy carbon, carbon-13. Carbon-13 has an extra neutron on it, and it is stable, so unlike carbon-14, which progressively loses extra neutrons, and that's used to like age-old archaeological objects, carbon-13 is a stable isotope, so it doesn't change over time. And it allows you to ask some really, really interesting questions. So I want to go back to these maps of the Galapagos just to show you again the temperature gradient, and these are all the locations at which I did censuses of fish communities, and I also collected fish to look at not only how does the community change, but how do diets at the species level change. And again, this is the sea surface chlorophyll, so it gives you a sense of how much variation there is in primary production around the archipelago. I'll say temperature has a more wide range of values, whereas with chlorophyll, it's like you're either in the west or you're anywhere else in the archipelago. So Marchena Island, that's MAR, this one on the top right, is very tropical. Median temperature is about 24 degrees centigrade, full of corals, lots of tropical fishes. It feels like you're in the Keys, basically, on a coral reef. In contrast, Fernandina Island, which is here in the west, less than 200 miles away, about 18 degrees centigrade, long-term average sea surface temperature, a lot of algae, a lot of filter feeders, the iconic marine iguana, the only marine lizard in the world feeding underwater on algae. So a totally different ecosystem. I think to get this same span of temperatures along our US Atlantic coast, you basically have to go from here up like past DC to get that same span of temperatures. So with that context, we did collection trips in July of 2019, which corresponded to the warm season, and then in January of 2020, which corresponded to the cold season, so we could cover some of the seasonal differences as well. This happened right before the pandemic, so I was able to get my samples and get out of there, and then Woods Hole, the lab, closed down for the pandemic for about a year. So we're actually only just now getting some of our data back, but it's really cool. So here's what we're seeing. This is all 25 species of herbivorous fishes that we found feeding on the benthos. So we actually put out cameras and recorded every single bite on the benthos and who did it. And I've separated the sites by kind of region. So there's the north, which is very warm. There's the west, which is very cool. And then central north and central south, which are kind of intermediates. And we had 11 different families of herbivorous fishes. You don't need to look too much at the species, but what I really want you to look at is that most species have higher herbivory rates in the north than anywhere else. And in fact, a lot of them were only found in the north. These are like tropical species, surgeonfishes, parrotfishes. And then some species actually have a different trend. So if you look at the bottom right, Stegastes beebe, it had its highest foraging in the west. So that's actually the opposite trend to what we would predict if temperature was directly limiting benthic herbivory. So again, we want to know kind of like, how does this fit into like global patterns? Well, so is herbivorous fish abundance and richness varying with temperature? Well, yes, it is. Both have linear positive correlations with temperature in similar ways that we see at the latitudinal scale. So that suggests that there is some underlying mechanism that's driving these patterns. It's not just, assembly or something or random. Even within the microcosm of the Galapagos, we see a very strong link between temperature and fish herbivory. But I wanted to test this hypothesis that if you get colder temperatures, the same species might actually have to stop eating algae and eat something else. And so that's where we get into the stable isotope analysis. So I mentioned these essential amino acids. These are proteins that we as animals cannot synthesize ourselves. That's why they're called essential amino acids. You have to get them from your diet. So since they are not synthesized by our bodies, they get transferred from one trophic level to the next without any alteration of their structure. And that provides a very, very faithful and reliable fingerprint of all the sources of primary production, all the different plants that photosynthesize, that capture energy and then transfer that energy into animals. So all food webs are flows of energy from the sun into plants and then into animals. And so we can actually track that flow of energy up the food web using this tool. So again, we're looking at these principal coordinates analyses. So it's like taking all 5 of those essential amino acids, treating them as axes. You got an X, you got your Y, you got your Z, then a W. So all these different axes are existing in the same hyperdimensional space, but we want to look at them in what makes sense to us. So we condense that information into 2 axes. And again, points that are more close to each other are more similar, points that are farther apart are more different. And first what I did was plot all of the points that correspond to the different sources of energy into the system, these primary producers. There's kind of 4 groups in the Galapagos. There's phytoplankton, there's corals, which have photosynthetic algae that live in their tissues. There's algae, which is growing on the benthos, and then there's detritus. Now detritus is kind of a catch-all for all these different sources of primary production that then get entrained into microbial mats on the seafloor, but microbes, unlike animals, can resynthesize their own essential amino acids, so they actually change that signature. So we've got our template of primary producers. Then what we do is we map on top of that all of our fish, and we can see where each species of fish is getting its nutrition from. Not just what it's eating, but where is the energy that it's getting coming into the food web. And so, again, you don't need to pay much attention. This is just kind of showing the spread of species. We've got a good mix of different sources of primary production. We've got pretty good coverage of the species within the realm of all the different primary producers. And if you look at these symbols, these are representing high and low upwelling regions, which is the cool west and the warm east, and then cool and warm seasons. So we did each region twice. And I'm going to relabel those, or I'm going to ask you to relabel them in your mind, as we're kind of going from the lowest temperature, highest production to the highest temperature, lowest production. So there's kind of 4 levels of temperature and production. But I'll walk you through that as we go along. So what I really wanted to get at was, let's test this hypothesis. If we look at the same species across a temperature gradient, is it changing its diet? Can it no longer feed on algae because it's too cold? So for that, we look at generalist species, species that have shifts in their diet across this temperature spectrum. So I'm going to show you 3 of those now. The first is Holocanthus passer. This is the king angelfish. And we've got, again, our 4 sources of primary production. And we've got our 4 different collection periods that we did this at. So I'm going to take you from the high upwelling cool location, that's the west during the cold season, all the way to the low upwelling warm location, that's the low upwelling northeast during the warm season. Those are our 2 extremes. So we look for the squares. Where are they? Smack dab in the middle of the planktonic production source. So that means that at the high upwelling, low temperature condition, this angelfish is feeding exclusively on planktonic sources of primary production. If we go to the opposite, the diamonds, it's smack dab in the middle of the detrital pathway. So it's getting all of its forage from detrital mats on the seafloor. So, and the 2 intermediates, the high upwelling during the warm season and the low upwelling during the cold season, kind of fall out right between those 2 extremes. So that tells me that the spatial gradient that we have in the Galapagos mirrors the temporal changes that we see with seasons and El Nino and La Nina, which I think means that this could be a pretty useful space for time proxy for long-term climate change. If we want to understand how future oceans will function in a warmer world, this system would be a relevant one to look at. If we look at Prionurus laticlavius, this is a surgeonfish, we see a similar change, but instead of going from plankton to detritus, it goes from algae to detritus. And contrary to the hypothesis, it's actually feeding on algae at the coldest and highest upwelling regions and locations. So this flies directly in the face of what we predicted, that it would be feeding more on algae in warmer conditions. That tells me that these species are not very close to their thermal maximum on either side, but whether it's cold or warm, and that they're actually responding to resource availability as opposed to responding to temperature change. So algae becomes more available during cold seasons because there's a lot of nutrients in the water, and the algae grows up from the benthos, just as phytoplankton does in the open water. And so this is a resource change as opposed to a temperature change effect. But it's challenging to disentangle those two things because they co-vary in the Galapagos. Temperature and primary production are so tightly linked. So future experiments in lab settings where we can manipulate temperature and nutrients separately would help disentangle that. Stygaces bb is the last one I'll talk about. This one similarly focuses on plankton in one condition, but then switches to this mix of detritus and algae. And it's got less of a common theme to it, more like a circular pattern. But what I'm seeing with both Stygaces bb and Prionurus laticlavius is that during the cold season, there's less differences between the two regions than during the warm season. And so that tells me that during the cold season, there's kind of a tendril, if you will, of water that comes from that upwelling center in the west and kind of wraps around the north and south and provides primary production bursts to the entire archipelago. So that kind of homogenizes things. During the warm season, there's some residual level of upwelling in the west, but throughout the rest of the archipelago, it's like null. And so that creates the biggest difference between regions. So that's pretty much all the research that I'm going to talk to you guys about today. I started at the Nature Coast Biological Station, which is in Cedar Key on the Gulf Coast, about the same latitude here in August of 2022. And I'm developing a lab. I've got some grad students that are coming in this fall and are going to be doing some similar work to this, but in the Gulf Coast. You guys probably know this, but we're located at a really important place latitudinally here because it's the lowest southernmost point at which the frost line occurs. And so there's a lot of tropical species that are kind of fixed south of that line because they cannot survive freezing temperatures at all. A couple of them, mangroves, are expanding northwards and actually are displacing salt marsh. Snook, which historically was found south of Tampa in the Gulf Coast, is now all the way up into Apalachicola. So this is an important transition point where tropical species are slowly but surely displacing subtropical species. So similar work to what I've been doing in the Galapagos, I think, could really be applied here. So with that, I'd like to thank all of you guys for coming, and I'll take any questions.